Hello, my name is Casey Edwards. Here's this, if you can see it. There we go. Casey Edwards, January 3rd, 2026. Okay, so today we're talking about the movement of macromolecules. Cells produce different macromolecules using distinct intercellular pathways, depending on the structure and the function of the molecule. And then I'm going to show you three flowcharts, and it shows the synthesis and movement of three macromolecules, which is a steroid hormone, cortisol, the protein hormone insulin, and intracellular protein actin. Okay. So here is the first flow chart. We're going to talk about cortisol. So steroid hormones such as cortisol are lipids, and they're derived from cholesterol and are synthesized in the smooth endoplasmic reticulum. So that's box number 1. And then enzymes in the smooth ER convert cholesterol into cortisol through a series of modification steps. And because cortisol is lipid soluble, its synthesis does not involve ribosomes, transportation vesicles, or processing by the Golgi apparatus. So after synthesis, cortisol diffuses freely through the cytoplasm and crosses the cell membrane by simple diffusion. And then once released from the cell, it enters the bloodstream and travels to target tissues, which in this case it binds to intracellular receptors that regulate gene expression involving metabolism, stress response, and immune regulation. Yeah. Okay, the next one is protein hormone, insulin. And so, let's go back. There we go. Insulin. So, protein hormones such as insulin follow the endomembrane pathway for secretion. Synthesis begins at the ribosome. Here we go, it's box number 1, which binds to mRNA encoding the hormone. Because the growth hormone is designed for export, the ribosome attaches to the rough ER, the rough endoplasmic reticulum, and the growing polypeptide enters the ER lumen. The protein is then enclosed within a transportation vesicle, and it buds off from the rough ER and carries the protein to the Golgi apparatus. Within the Golgi, the protein is modified, folded, and sorted, and then after processing, it is packaged into a secretory vesicle, which move towards the cell membrane. And these vesicles fuse with the membrane and release the hormone by exocytosis, last box here. And once in the bloodstream, it acts to let the cells know whether to take in more glucose or not. Okay, last is actin. It's an intracellular protein. So intracellular proteins such as actin remain within the cell and do not enter the endomembrane system. Actin is synthesized on free ribosomes in the cytoplasm. So just floating ribosomes, not in the rough ER. And after translation, actin monomers fold into their functional shape and assemble into filaments. So that's this step right here. And these filaments form part of the cytoskeleton, where they provide structural support, enable cell movement, and participate in processes such as muscle contraction and intracellular transport. So the final destination of actin is within the cytoplasm where it is made, so it functions as a structural protein rather than being secreted. And altogether these three pathways demonstrate how cells use specific organelles and transportation vesicles to ensure that macromolecules are delivered to precise destinations and perform their intended biological functions. And I have here my references page. I use 3 different references. And once again, my name is Casey Edwards. It is January 3rd of 2026. Thank you so much.